Good afternoon. I have the pleasure of introducing Dr. Gregor Bucher as our next speaker and also as a new member of our faculty. Professor Bucher studied zoology and developmental genetics at LMU Munich, Germany, where he studied the function of patterning genes in the red flower beetle and discovered parental RNAi. After 2 years of childcare and working as a scientific writer, Dr. Bucher established his own independent research group in Göttingen. In 2013, he was awarded a Heisenberg professorship, and since 2017, he has been a professor in the University of Göttingen. He's interested in the genetic basis of development and evolution of insect head and brain and new modes of pest control using RNAi. Please welcome him with a round of applause.
So thanks a lot, especially for the organizers for giving me the opportunity to present my data here. Although it's not about protein or protein complexes or cells, it's really about organs. It's about brains, and not from vertebrates or humans, but from insects. I hope you still enjoyed this kind of adventure into evolutionary developmental biology. So the starting point is this here. What we observe if we look into nature, the animals have very different brains. Here, for instance, the vertebrates, all the basic setup, but they are different. For instance, the lion here and the gibbon here, these brains have to do some different task in the same situation to survive. So apparently, despite having the conserved basic setup, they have to function in a somewhat different way. And the same, of course, is true for insect brains. All of them have eyes, antennal lobes, and central complex, mushroom bodies, but they all look different. And these adaptations are probably very important for the survival of the animal. For instance, what we see is that some brains are really big, so these are to scale, right? So this brain is several-fold bigger than this one. And then also the respective neuropiles are different. So, for instance, big eye lobes here, small eye lobes here, or small mushroom bodies here, big mushroom bodies here. So not only the overall size of the brain is diverging, but also the subparts of the brains have adaptations. And these adaptations are really of evolutionary significance. For instance, bees here, they have very big mushroom bodies, and the mushroom bodies are those structures that are required for memory and learning. And they do that very well, right? The red flower beetle, with which I'm working on, has rather small mushroom bodies. Probably it's not such a good learner. It's burrowing in the ground, so it has very small eye lobes, while these two guys here are navigating in the environment and space, so they really need good eyes and also big processing centers for eyes. So this divergence that has been well described in insects is of course adaptive and probably very important for evolutionary success. And of course this divergence emerges during development. So we have to understand the divergence of development if we want to understand the diversity of morphologies that we have. While the diversity of brain shapes and sizes is very well described, it's completely unclear what the same cells do in different insects to form different brains and what the genetic regulation of that might be. And that's, let's say, the basic answer, the basic question that we're asking in the lab. One example here, the central complex is a higher order brain center important for navigation, and all insects have it, of course, but you can see that the respective subparts in the different insects have different shapes and sizes, but the underlying connectivity pattern is very similar in all of them. And there's a very curious developmental divergence with respect to the central complex. In grasshoppers, for instance, the entire central complex develops during embryogenesis. So the first hatchling already has a fully developed central complex with all aspects, all connectivity that it needs. It just grows over time, and the adult has just a bigger version of the hatchling. This is very different in Drosophila. If you look at that, you have it in the adult, but before you don't see any sign of a central complex. What you do see are some cells that cross the midline that later form the central complex, but there's no synapsin, no connectivity, nothing. And the beetles that we are working with actually are something like in between. This is called heterochrony, like a difference of developmental timing. And the beetles with which we are working is something like in the middle. So we have, during embryogenesis, the formation of a partial central complex. And with that partial central complex, the larvae is orienting itself in the environment. And only during metamorphosis, the development is completed to form the full central complex of the adult. And it's completely unclear what the same cells do if we compare this situation, this situation, this situation. And this is something that we wanted to ask. But of course, to compare hundreds or even thousands of cells in dozens or hundreds of different species is just too much. So we have to simplify our model system in order to really be able to study that. And the first simplification is that we just say, okay, let's compare two insects with each other. This is difficult enough, and let's expand afterwards into more species. Of course, we're using the main model system, Drosophila melanogaster, as the main model system because it's such an amazing model system. There's most knowledge about gene function is gained there, so that's an excellent point of reference. And for comparison, we use a red flower beetle, which in terms of gene function tools is something like the second best model organism in arthropods. And I've shown you already the differences that there are. Development of the nervous system is very similar in all insects, basically. So development starts with a neural stem cell dividing asymmetrically into a ganglion mother cell, which divides once more to form postmitotic neurons. And while these neurons then start growing out axons, the stem cell continues to divide, forming another ganglion stem cell, which will divide to form another pair of cells. And at the end of embryogenesis, a group of cells emerging from the same stem cell forms something like a group of cells with a bundle and also similar, not identical, but similar connectivity. And the stem cell goes into quiescence and waits for later stages. And this is essentially the brain with which the first larval instar is living. During late larval stages, neurogenesis starts again, and so the same neuroblast starts again dividing. Additional cells are formed. They use the scaffold formed by the embryonic cells for their axons. And then in the end, we have the adult connectivity with embryonic and post-embryonic cells, and the stem cell will eventually die. In principle, this you will find in all insect embryos. So if you look at Drosophila, you would in principle find the same. And with respect to the central complex, we have, let's say, the same end structure, the central complex. We have the same starting point, the dividing stem cell. But we don't know what the difference is that explains this difference. For instance, it could be that the stem cell is not dividing in the embryo but waits for later. It could be that it does only some divisions and then stops to continue later, but it could also do all the divisions and maybe just form axons without any connectivity patterns. Still, comparing two insects is still very complicated because each larval brain has more than 10,000 neurons, so this is still too much, so we have to simplify again. And then what would be really nice is if we would not have to look at all neurons of the brain, but look only at a subset of it. For instance, by genetically marking, let's say, one or several stem cells or subsets of neurons with GFP, for instance, this would simplify our work, because then we would not have to look at all the cells, compare them, but we would only look at the few marked cells and compare their development. So the question is, how can we mark homologous cells in two different species? This is not so easy. You can mark randomly cells in one or the other, but to say I have homologous cells marked into different species, this is not such an easy task. So the second simplification that we're doing is to mark subcells and or the subparts of cells. And the concept that we have introduced here is a genetic neural lineage, which means or is defined as all cells that are expressing one highly conserved developmental transcription factor. What do I mean with highly conserved transcription factor? It is known that the anterior development of all animals is based on a set of 1 or 2 dozen of conserved genes that you will find in humans, in insects, in cnidarians at the anterior part. And these are some examples. So left half is the anterior neuroectoderm of insect, the right half of a vertebrate neural blade. And you can see that the same or homologous genes mark more or less similar regions in the early neuroectoderm in these 2 clades, meaning that, for instance, Rx here in green is marking an anterior-most neural part in all animals. And we think that if a transcription factor is specifying the anterior-most part of the brain in mice and in insects, then it's very likely that this transcription factor will mark very similar or homologous cells between 2 holometabolous insects. And so we have used this gene as marker. We could have used any other of those as well. And wanted to compare only the cells that express that transcription factor. One way, of course, is doing this by antibody stainings. So we had one from Drosophila and we generated one from Tribolium. You can really see in that brain with the eye lobes, with the central complex being here, you see these cells in a lateral anterior region, in a dorsal median region, and so on. And you see that in both species. But of course, if you want to analyze the brain, cell body location is not what you want to know. It's only part of the information. The really important information is the connectivity pattern of these neurons. So how do we mark homologous neurons with their connectivity? And to that end, we have used homology-directed repair in order to generate bisystronic lines. The principle is shown here. We have a gene, very simplified structure. We have a coding region, a core promoter, and an enhancer. And we would like to mark these cells with GFP. So what we did by genome editing is to expand the mRNA sequence with GFP. And we separate these 2 coding sequences with a 2A peptide, which leads to ribosome skipping. So one ribosome is making 2 separate proteins from one messenger. So upon transcription, a messenger RNA is formed which is extended by the GFP and 2A sequence. And after translation, we have 2 separate proteins in a more or less stoichiometric relationship to each other. And that's important that we don't have a fusion protein because Rx is localized to the nucleus, and we want GFP to be cytoplasmic in order to be able to mark the projection patterns of neurons. In our experience, this technique is a bit cumbersome, so it really takes a lot of time until we have the construct and the transgenic line. But once you have it, it's really very precise, much preciser than enhancer traps or other tools that we have tried. So this is the way to go. And now we have 2 insects, and in both of them we have the identical bisystronic construct, meaning that all Rx positive cells are marked by GFP. And these are the pictures of these brains. And we checked, of course, with the antibody, and the thing is 100% overlap. So here you see a scan through the brain. You see cell bodies, you see the projections in the central complex. Another bunch of cell bodies coming here. And if you now go to the next one, essentially the same you see in Tribolium. You see cell bodies, the central body marked, and then again another bunch of cells occurring here around the protocerebral ridge. And now we have this tool which is kind of simplified and allows us in more detail to study the divergence that we know of. And now with these tools we are now able to get first insights into what the cells do in a different way during that heterochronic development. Max Vonroth in the lab, he started that project and he tried to answer, got first insights into that question by giving names to the different stages of development. For instance, the first axons crossing the midline, the first formation of these certain cell groups in a typical arrangement with this perpendicular arrangement, the first decussations, these X, Y things, and so on. So he defined 15 different stages of development, and then he looked in Drosophila and in Tribolium when these stages would occur. So he really did many stainings in Drosophila embryos and Tribolium embryos to really compare when he would find a certain stage. He did that also in several larval stages and also several pupal stages and also in the adult. And based on really a lot of data, he was able to draw a very complicated scheme. I won't go into detail here, just the principle. So here we have embryonic development from 0 to 100% embryonic development, then larval development from 0 to 100, pupal development from 0 to 100, and here we have the developmental steps from 1 to 15. This is a bit too complicated to go through, so I have simplified it a bit. What we see in the grasshopper, or what others have described in the grasshopper, all these events occur during embryogenesis. The late stages of embryogenesis start expressing synapsin, so then we see some functionality, or we can assume it, and the hatchling already has a fully developed central complex and maintains this to the adult. In flies, only the first three developmental stages are similar. The fourth one is shifted in time into post-embryonic development, but still non-functional, so we don't see any synapsin here. And during metamorphosis, the remaining steps are done. And this shift in timing, as I told you before, is called heterochrony. In Tribolium now, the first steps are again in embryogenesis and are a bit shifted. And number 4, the first difference is this number 4 is already probably active because it is expressing synapsin. So we can assume that there's some neural activity here, but not here. You would expect now number 5 to occur here, but what we found that number 5 is actually before number 4. And this is called sequence heterochrony, when a sequence of developmental events is kind of switched, and then the rest is occurring. So now what we now know is that this partial central complex that we find in Tribolium is really actually a developmental stage, because morphologically it is identical or very similar to the stages that we found in flies and in grasshoppers, but it has become functional by the expression of synapsin. This kind of says this is not some special thing, this is not a special reduced central complex, it's just a developmental stage that became functional. So we have now very well described this, but we don't yet know about the genetic control. That would be the next step. So this was, let's say, the first dimension of brain diversity that we have started using this tool, and now we're coming to briefly to a second one, for the genetic control of adult neurogenesis. So now we're talking about these structures here, the so-called mushroom bodies. I've shown you that with a bee that has very big mushroom bodies for memory and learning. And the difference between Drosophila and Tribolium is that these mushroom bodies, after eclosion from the pupa, there's no more neurogenesis in the fly. But in beetles, we have ongoing neurogenesis in the mushroom body. So there's a difference in stem cell activity between these two species. And by chance, the Rx genetic neurolineages is marking the mushroom body stem cells. So in embryos and in larvae of both species, we find the mushroom body neuroblast being marked by Rx. and so are the most recently born neurons from the mushroom body. So that's the same in both. In the adult beetle, you still see the two mushroom body neuroblasts on each side being active, expressing Rx and dividing and forming neurons. But in Drosophila, this is lost. So you don't find these, you don't find Rx expression, and you don't find the stem cells anymore. And it has been shown in Drosophila by these guys here that Rx expression is actually required for maintaining stem cell activity, and if you knock down Rx precociously, then also the stem cell will die precociously. So now the model is very clear. So we have Rx expression in these neuroblasts, which drive stem cell activity, like dividing activity, and this leads to neurogenesis throughout stages. But after eclosion from the pupal case, Rx expression goes down, stem cell activity goes down, the stem cells die, and we find no adult neurogenesis in flies, but it's ongoing another weeks or months in tribolium. And this is now such a specific phenotype that we kind of might have the chance to really find out an enhancer element in this one gene, in the Rx gene, that in one species is reacting to hormonal signals in a different way than in the other species, explaining the difference in expression, explaining the difference in stem cell activity and adult neurogenesis, and if you're really lucky, we will even find differences in learning behavior or learning capacity of freshly hatched beetles compared to flies. But this is all kind of something that we're currently working on. We don't know yet if this is going to happen. Now I come to the last dimension of diversity that we can study very well using that lineage, and that is that we found a cluster of cells, of neurons that we find in the adult Drosophila brain, but not in the tribolium brain. And this is of course very curious. If a brain has an entire cluster of neurons in addition to the others, this can really relate to some different function. But we have no idea whatsoever, right? But this is also something that this comparing subsets of neural cells between species allows to do. So we find these three clusters arranged in a line in both species, also with similar projection patterns, and then here we see this, and this gives us a handle on studying the diversification of brains. Okay, so how are we going to continue from here? First, with respect to heterochronic development of the central complex, now we would like to find out what the genetic control is that allows development to a further stage in one species and stops development in the other. And the main candidates are, of course, the ecdysone and juvenile hormone pathways, which regulate many processes during embryogenesis. We would also like to know what actually is the difference between a larva which does not have any central complex. So it does not have a compass in a way, it does not have means to navigate, while this guy has. So what is the difference? The hypothesis would be these guys here are living like in food, so they don't need to navigate. They're in the rotten fruit, so they don't need to find anything, while these guys here really have to find food and have to run for them, so they need to navigate. But that's just a hypothesis. With respect to adult stem cell activity, as I told you before, we're trying to test the hypothesis that I have laid out. And with respect to this divergent cell cluster, we're now starting we're doing the first single cell sequencing of these transgenically marked cells in the hope that we really find what the molecular signature of this additional cluster may be, and then using the power of the Drosophila model system to find out what cells are these, where do they project, what are they doing in behavior. And of course we would also like to know what is the genetic regulation that allows to form an additional cluster here in flies but not in beetles. Something that I have not talked about, I would just briefly touch upon, are other kind of aspects of brain development that we're studying. One is this: we know that all insects start their development with stem cells, of course, but beetles have, let's say, 120, 130 stem cells in the brain. Flies have only 80 or 90. So we would like to know, are these completely different stem cells that beetles have, or just more of the same type? And Noel has done really great single-cell sequencing of beetle neuroblast and has got comparative data from colleagues that have done the same thing in Drosophila. And now we're trying to find out if we can find out different types of stem cells. I've shown you that we have to simplify our system, and using the genetic neural lineage really has simplified the system from several tens of thousands of neurons to a couple of hundreds, which is still a lot. So we are still working on further sparse marking of the brain to have really more precise imaging. The classic thing is rainbow, disadvantage being that it's a random system, so you cannot control it, but we're kind of trying to devise tools also by genome editing that allow to mark the same neurons in different species. And with that, I would like to thank the team. Much of the work that I presented was initiated by Max some time ago. Georg has done tremendous work really comparing in much detail these two lineages. Christine, she is now continuing that work, trying to hunt down the stems, the adult neurogenesis stuff, and talked about Noel and Ying with their projects. With that, I would like to thank you for your attention.